Okay, so this is the first chapter in Unit 4, right? Okay. So chemical communication, this started to be a small chapter and now it's getting too large. Anyway, chemical messengers, things like hormones, neurotransmitters, prostaglandins even. And so they bind to the receptor proteins by the lock and key and induced fit. Some enter the cell directly and some need a second messenger. And this is how we control metabolism, is by controlling the enzymes. So chemical messengers can either activate or deactivate enzymes, activate, deactivate genes that produce proteins, including enzymes. They can affect the permeability of membranes, or they can cause the formation of a second messenger. So I kind of reorganized this chapter on the basis of 6 classes of messengers. Some we know a lot about, some we don't know that much about. But your brain is only 2% of your body mass, but uses like 20% of your energy. Nerve transmission, 268 miles per hour. So anyway, here's a picture of what they presume is most of the connectosome. It's lost some resolution, but manipulating the image has lost some, but you get the idea. So we have the central nervous system, we have the peripheral nervous system. And the central nervous system is the largest part, peripheral nervous system. And this is not a neurology class, this is not a physiology class. So what we're really interested in is in biochemical mechanisms. Although I did feel compelled to kind of give you a good idea about how nerve transmission works. So the peripheral nervous system, you have the sensory and you have the motor. And so here is the central nervous system, here's the peripheral nervous system. And you have receptors over here that sense things. So this is a sensory neuron. So it senses touch, temperature, things like that. And then it goes through the central nervous system, including the brain. And then there's a response to the environment. So here you're sensing the environment, and here you're responding to the environment. So you move your muscles or whatever it is. So the motor division is either somatic or visceral. So the somatic part is under voluntary control, and the visceral part is involuntary, also known as the autonomic nervous system. And here's kind of a good scenario, okay? So here's the central nervous system, here's the peripheral nervous system, so they communicate both ways, right? And so if you look down here, here's the sensory, here's the motor. So the sensory sends information up here, then it responds with the efferent or motor part. And here's the somatic, and here's the autonomic. So this you can control, this you cannot control. So what you cannot control mobilizes different things during the fight or flight syndrome or whatever. This one basically conserves that, right? So this mobilizes, this conserves energy. And of course, it's all connected, okay? So we went through the structure of a lot of drugs in chapter 25 because many of them are amines. And so again, an agonist is something that competes with the natural messenger or the receptor protein. Antagonist blocks the receptor protein. And then one thing that's kind of interesting is that people respond differently to different drugs. And even when they do respond in a similar way, they may need completely different dosages. So what you might explain this with is what's called receptor promiscuity. So everybody has a different set of genes that are either upregulated or downregulated depending upon what their environment has been. And so, for example, I'm using smell. So one person may have 80% of receptor A and 20% of receptor B. Another person may have the opposite of that or something totally different. So if receptor A binds a drug more efficiently than receptor B, the person with 80% receptor A is going to respond more strongly to the drug. So anyway, this is the reason why we have personalized medicine. This is the reason why we have doctors prescribing different amounts of drugs. And so here's kind of an idea of this. So here's the olfactory bulb. This is what your nerve tissues in your nose respond to. But this is the collection. So that goes to the central nervous system. But here's where the olfactory odorants are. Here's some cilia. Look, these are nerve endings right here. And they contain receptor proteins. So here's the receptor proteins. So here's the different kinds of odorants, okay, and the description of them. So here's the rancid, sour, goat-like smell. What would happen if you didn't have receptor 5? Well, then you wouldn't be able to smell those things. What happens if you had receptor 2 and you didn't have receptor 6? You would smell sweet and herbal and woody smells differently than someone who had both of those receptors. Anyway, I think that gets across the idea. So the first messenger is an extracellular chemical. It's either a hormone or a neurotransmitter that binds to the receptor on the target cell's surface and causes a change in metabolism. So the second messenger is a chemical such as cyclic AMP that we saw in the previous chapter. So we're going to look at a few systems. We're not going to look at them all. And I'm trying to figure out a way so you're not having to memorize a whole bunch of stuff. I don't want that to happen. So what about hormones? So there's a stimulus. The environment changes. A neurotransmitter goes to the central nervous system. The central nervous system sends a transmitter to the hypothalamus, for example, which is the major endocrine gland. And that hypothalamus sends a hormone to another endocrine gland or directly to specific tissues. Okay? And prostaglandins are not true hormones because they really affect the same tissues that produce and secrete them. So these tissues are making their own. That's what they're doing. So steroid hormones are hydrophobic enough to pass through the membrane. They go right into the nucleus and affect genes. Okay? Either turn on or turn off a gene. So those are hydrophobic, and the hydrophilic ones, the water-soluble ones, are either peptides or amino acids. Sometimes they're things such as cyclic AMP. So these give you short-lasting effects because they don't affect genes. So the neurotransmitters, they can be cholinergic. They can be adrenergic. In other words, anyway, let me find this figure over here. Here we are. So here's a typical nerve. So here's the nerve fiber, the axon. This one is coated with sheath cells, okay, to protect it and to increase its transmission. So here's the cytoplasm, the nucleus, the cell body, the dendrites. So here's the synapse between two different ones. So this one right here could be a sensory neuron. Then that one connects to some other nerve fiber. So here's some of the major signaling systems. So we'll talk about G protein, second messengers, and then here's some of the systems that's involved in. So the cyclic AMP system, the phosphatidylinositol system, we'll look at that. I'm not sure if we look at this one. We'll look at some cyclic GMP. We'll look at a few of these. Okay. But I'm more interested in you getting a general idea than you having to memorize a bunch of things. Okay, hold on. Here we are. So acetylcholine. Let's see, figure 2, where did that go? I have my figures messed up. Oh, here's figure 2. Okay. Yeah, that is what I'm looking for. Okay. So here's the acetylcholine system. So here's this acetylcholine receptor protein. And you see that it requires 2 molecules of acetylcholine to bind to it before it opens up. So what does it do? Well, if this opens up, sodium, which is at a higher concentration outside, rushes in. Potassium, which is a higher concentration on the inside, rushes out. And so you get a depolarization of the membrane, because out here is positive, out here is negative, because of unequal distribution of charges. So that is what the depolarization event is. So when it depolarizes, that signal comes down over here. So you see how it's positive on the outside, negative on the inside, because there's more sodium out here. And then here's where the depolarization is traveling down. So they're showing you the depolarization right here. In this particular case, it also causes calcium to be released into the cell. Here they're showing it to you externally, but it can also be internally held in own little vesicles in here. But regardless, calcium is required for the process where it takes these vesicles full of acetylcholine and it causes them to fuse with the membrane. When they fuse with the membrane, their contents are released into the synapse. Okay? And these are the acetylcholine receptors. So that's how you get the signal from one cell, one nerve cell, into the next nerve cell. Wait, was that being shared? Sorry, that wasn't even being shared. I'll have to do that over again. So as I was saying, you require 2 acetylcholine molecules to open up this channel. So here you are in the resting state. So here's the channel's been open and the depolarization is traveling down here. And that causes calcium to come into the cell, causes the fusion of these vesicles that contain acetylcholine with the membrane, and then release the contents into the synapse. Then the acetylcholine binds to the receptor proteins on the next cell, causing a depolarization to the next cell. So here's the scenario, 1, 2, 3, 4. Calcium here is acting like the second messenger. And then finally, the acetylcholine has to be hydrolyzed because you don't want that signal to just sit there being, turning on nerve cells. The lifetime of the acetylcholine and the synapse must be very short. So they're immediately destroyed by acetylcholinesterase. If you want a larger signal to go to that next nerve cell, you have to have continuous excitement of the presynaptic cell. Okay. So this way, the response of the nerve is proportional to the extent of the environmental change that you're responding to. So here's a couple of things that affect this system over here. Botulism toxin is probably the most potent toxin known. It prevents the release of acetylcholine from the storage vesicles, so you don't get any nerve transmission. Many nerve gases inhibit permanently acetylcholinesterase, so the neurotransmitter acetylcholine has an indefinite lifetime. So nicotine at low dosage is a stimulant because it acts as an agonist and prolongs the action of the nicotinic acetylcholine receptor protein. But at higher concentrations, it binds to the receptors as an antagonist, blocking the action, which is why nicotine is extremely toxic at high concentrations. So why is nicotine addictive? Because it allows the release of dopamine. That's the neurotransmitter in midbrain neurons. And that's what gives you a feeling of satiation, of relaxation, and things like that. So amphetamines and cocaine also act at those same receptors and also cause the release of dopamine. So acetylcholine synthesis is inhibited in Alzheimer's patients. So one thing they do is they use an inhibitor of acetylcholinesterase to increase the amount of acetylcholine in the brain. Competitive inhibitors of that same enzyme can be used as muscle relaxants. And what do local anesthetics do? They block the acetylcholine receptor and prevent transmission from the sensory neurons. General anesthetics work completely differently. They go to mitochondria and they inhibit the synthesis of ATP. Insufficient ATP reduces the ability of the nerve to get an excitable membrane, so therefore it cannot transmit nerve transmission as well. So the adrenergic messengers, the monoamines, also known as catecholamines, these are things like epinephrine, norepinephrine, serotonin, dopamine, histamine, things like this. Basically, we talked about them in chapter 25. And in chapter 30, we talked about the fight or flight response. So that has to do with epinephrine, right? And so here's some of the effects So inhibitors of monoamine oxidase prolong the effects of monoamines. So MAOs are monoamine oxidase inhibitors. And when you inhibit that, the amines last a little bit longer. So those are used as treatments for for depression and for hypertension. Dopamine is an excess in schizophrenia. Amphetamine abuse can result in schizophrenia. I was just talking to one of my colleagues yesterday about this. She's from Korea, and she's been in the United States about 6 years, I guess. And we're talking about the different ways that the countries take care of homeless people, and she's just surprised that homeless people are walking around doing whatever they want in Honolulu. But anyway, we were talking also, it's like, how many of those people were turned schizophrenic? They might have been pre-schizophrenic, but they were pushed over the top by drug abuse. I'd say it's a very high number. So we have talked about histamine before. Tyramine is produced by the breakdown of the amino acid tyrosine. It's found in some food. Some people are extremely sensitive to tyramine. It gives them headaches, nausea, and all kinds of stuff like that. So anyway, if you are on monoamine oxidase inhibitors, then you should be very careful about that and watch what foods you eat. Let's see. So here's tyrosine right here. Here's one step over here where you start to produce tyramine. Okay. Tyrosine, the amino acid, is also used to make dopa, and then dopamine, then norepinephrine, and epinephrine. So here's some amino acid messengers. Glutamate. So glutamate is a major excitatory neurotransmitter. So what it does is it enhances the excitability of neurons. The most abundant amino acid, representing about 60% of the amino acids in your body. It can also be used to synthesize blood glucose during fasting or starvation. Okay. And glutamate can be converted into gamma butyric acid. So here's glutamine, here's glutamate. Okay, so look, it's a glutaminase enzyme. So what does it do? Here's an amide, a simple amide. So it cuts or hydrolyzes off the ammonia, and you get the glutamate. And you've heard of monosodium glutamate, right? Same thing. And then you have a decarboxylase that cuts off that the carboxyl group right here, and you end up with gamma aminobutyric acid. So alpha, beta, delta, gamma. Okay. And so while glutamate enhances the excitability of neurons, after it's decarboxylated, GABA actually reduces the excitability. So they have exact opposite effects. So this is kind of an interesting concept here. Here's the enzyme right here that converts between glutamate and GABA. So this enzyme is going to control whether your neurons are in an excitability state or a non-excitability state. You might ask, okay, well, what the heck is controlling glutamate decarboxylase? But something has to be able to do that, right? Well, it's metabolic pathways. So Here's a typical scenario right here. So here's glutamate. It's a neurotransmitter. So it gets converted into GABA right here. And then something happens where it starts to release GABA into the synapse and then causes some effect over here. Okay, well, what happens to GABA? Where does it go? Well, it gets sucked into another cell directly associated with the synapse right here. And the cell is called an astrocyte. It's like a helper cell. And basically it recycles it back into glutamine to be given back to the other nerve. So the lifetime of GABA in the synapse right here should be minimum. How do you get it out of here? Okay, with the astrocyte cell, the helper cell. Right. So some peptidergic receptors or messengers, glucose transport proteins. Let me get that figure up. Figures 1 through 4. I think I covered all that. Oh, here's figure 4 right here. So here's the receptor, here's the primary neurotransmitter. That conformational change right here causes a conformational change in the G protein. It activates G protein. What does G protein do? Well, it hydrolyzes GTP, which is similar to ATP. And what it does then is it goes and activates adenylate cyclase. Okay? And that's what produces cyclic AMP. So here's a case where a protein is a kinase. Okay? It's not active until cyclic AMP is in there. Okay? So when it does that, it takes and puts phosphate on different things. So here is grabbing the phosphate for a few minutes, right? And then when it interacts with this ion channel, it goes and transfers, oops, sorry. Wait for that to go away. So the phosphate gets transferred from the protein to this inhibitor protein right here that's blocking the channel. So when this gets phosphorylated, the conformational change happens and it opens the channel up. So one concept I'd like to get across, which is kind of my own concept, but it's important in terms of function. Right now we're looking at all these proteins on the membrane, and they all seem to be separate. But another way to think about it is, these are all part of the same enzyme complex. It's a huge enzyme complex where when an effector molecule binds to this subunit, conformational change here, causes that one to be active, conformational change here, conformational change here, and conformational change here. So all of these proteins right here are being used to regulate this ion channel right here. And they're all working in a cooperative way. Cooperativity between a huge multi-subunit complex. Okay, so let's see. Open up another one of these now. Got so many windows open on my computer. I'm gonna have to close some. But they're not letting me do that. Okay, not finding it. For some reason, not showing up. Okay, I'm gonna have to start closing things so I can find things. Okay, so this one is the insulin receptor. Now, insulin is a polypeptide, right? It's a protein. So here's the receptor sites for insulin. Here's the insulin receptor. Look at it, it's a multi-subunit complex of proteins, right? So when insulin binds to these two receptor sites, it needs them both to be bound. Otherwise, the cell won't respond to that stimulus because the stimulus is not strong enough. A conformational change here causes a conformational change here, which causes a conformational change over here, causing something to happen over here. Now notice that these are labeled as tyrosine kinase domains. So a domain is part of the protein. These, embedded in the membrane, are phosphotransferases. Okay. So what's happening over here is that this is a target protein. So I call it an insulin receptor signaling molecule or something like that. I forget what it's called, but it's the target. So the first messenger is insulin. The second messenger is going to be this target protein. So how does this work? You activate the protein kinase. It then interacts with the target protein, puts a phosphate on it and a tyrosine on it, and that causes the intracellular insulin effects. Okay. So why do they show this phosphate sticking onto the tyrosine kinase part? Because when this is all turned on, these guys take ATP and just start phosphorylating each other. So they become loaded up with phosphate groups. And when the target protein bounces into it, then that's transferred to that target and activating it. This is the second messenger. It's also a protein, by the way. Okay. So here's another scenario of that. Okay. So here's the insulin binding to the insulin receptor. Here's the autophosphorylation sites. What does the target protein do? Well, it can do several different things. If it goes this way, depending upon what protein is being phosphorylated, it activates what's called the MAP kinase signaling pathway. It promotes cell growth, proliferation, gene expression. If it goes this way, it activates the IP3 kinase pathway. What does that do? Well, it promotes the synthesis of lipids, proteins, glycogen. It can also do things to help cell survival. But here's one of the major things it does, and what people concentrate on. Depending upon what target protein is phosphorylated, it may go through this IP3 kinase phosphate pathway and end up activating the glucose transport proteins. So you know that the release of insulin from your beta pancreatic cells causes glucose to be absorbed by the cells, and thereby reduce glucose concentrations in the blood. You may not know that the glucose transport proteins are not even on the membrane unless insulin has been activated them. They are stored in a vesicle and they don't fuse and then combine with the membrane until insulin has been absorbed. And it goes through this pathway right here. So when insulin is low, the glucose transport protein is found in these vesicles, right? When blood glucose increases and insulin spikes, insulin binds the receptors. This goes to the membrane and you absorb glucose. So let's see. I don't know if I want to get into all this detail. Anyway, so here's your GLUT4, your transfer protein. Here's your insulin receptor. And you see it's quite complex over here. Some of the mechanism is not known. Most of the mechanism is known. What we do know is that exercise can activate the MAP-AMP kinase and cause this to happen. And I guess I went into much more detail than I wanted to. But anyway, so inositol triphosphate kinase is involved in this process over here, and it gets quite complicated. So there's a lot of things that can go wrong other than just this part right here. So two types of diabetes that we'll study later on. One type is that you don't release enough insulin. But another type is you don't have enough receptor proteins for the insulin. That's adult onset diabetes. But you should know right now that it's more complicated than simply the receptors. If anything goes wrong in any of these systems right here, you're not going to get the glucose transporter to go into the membrane, and you're not going to be able to absorb glucose. So adult onset diabetes is quite complex and involves all these different processes, and exercise is involved. What's one of the mantras that you keep hearing me say? Is exercise is extremely important, as well as everything in moderation. Okay. So anyway, what about some other ones? So substance P, or P protein, is a neurotransmitter and neuromodulator that causes pain. Okay, so what does that mean? Endorphins and enkephalins inhibit pain by inhibiting the release of substance P. Strenuous exercise causes the release of endorphins and enkephalins. So strenuous exercise makes pain go away. Capsaicin that comes from chili peppers causes the release of substance P from its receptors. And morphine binds to the same receptor as enkephalins and also inhibits pain. Leptin is a protein that suppresses appetite. It binds to its own receptor. But anyway, what is an interneuron? Okay. So here's the receptors on the skin. Here's the afferent. Here's the efferent. What is an interneuron? The interneuron is what connects those two processes. Incoming information, outgoing response is moderated by an interneuron. And that's essentially where most of, or many of these things act, okay? Right there at the level of the interneuron. And so another category that's been added are the neurosteroids or neuroactive steroids. So they could come from outside the cell or in the cell. And steroids, normally we think of them as long acting and slow acting, but there are this class that acts rather rapidly, okay? And they almost always affect brain function. So some of them also affect gene expression, like most of the steroids you would expect. But anyway, these have wide range of potential clinical applications, including sedation, treatment of epilepsy, traumatic brain injury, and things like this. But we don't have a lot of examples of those. Okay, so I think I should probably rearrange some of my figures. So I guess I need to review figure 4 here for a second. So let's just go over this again, because I think this is important. So here's a neurotransmitter or hormone, hits the target cell, causes a conformational change. Part of this active group of enzymes or proteins over here is GTP, which is an enzyme. It's inactive until it gets this conformational change that happened from the receptor binding to this other part of the protein. And it does take GTP and it hydrolyzes it. When it does that, it comes over here and activates adenylate cyclase by conformational change. What does that do? It produces cyclic AMP. That's what adenylate cyclase does. It produces cyclic AMP. So what does cyclic AMP do? Well, it goes over and it activates this protein kinase over here. Protein kinase is a phosphortransferase. So here's the catalytic subunit right here. Here's the regulatory subunit right there that causes it to be inactive. But when cyclic AMP binds to this regulatory subunit, it causes a conformational change, and this comes off. Now we have the active phosphoprotein or phosphortransferase. So what does it do? It essentially transfers the phosphate group from ATP to this regulatory protein on the actual ion channel. That causes it to go through a conformational change, which basically opens up the channel. So what we've done is this first messenger caused this channel to open. It didn't do it directly, it did it through a second messenger, which is cyclic AMP. I just realized that, again, that wasn't shared, but that was a review anyway. Okay. So now we're going to look at figure 6. Okay. So here's another second messenger system. So a hormone or neurotransmitter binds this receptor protein that activates G protein that turns on this enzyme called phospholipase C. Okay, what does phospholipase C do? Well, it takes this lipid right here and hydrolyzes it. So the polar part over here actually is inositol. So when you hydrolyze it, you get inositol triphosphate. You saw this in the chapter on lipids. So this is a water-soluble second messenger. It ends up opening up calcium channels. So calcium now rushes into the cytoplasm from storage vesicles. Okay. What does the other part do? This diacylglycerol right here goes and it activates protein kinase C. Why do they say protein kinase C? Because that's associated with calcium. Okay. And what does a protein kinase do? It simply takes a protein and puts a phosphate on it. So whatever protein is predominant in that cell is going to get phosphorylated. And it might turn from an inactive protein to now an active protein. That's going to go off and do cellular changes in metabolism. So really, this is a phospholipid in the membrane. It's stored in the membrane, and when we hydrolyze it, we get two second messengers. One water-soluble, one lipid-soluble. Okay. And the purpose, again, is that this is a way for that cell to change its metabolism and respond to the environment. So anyway, here I talked about G proteins a little bit. It's just only important that you know what a G protein is and what it does. Why is it called G protein? Because it binds GTP. Let's see, so some alkaloids such as caffeine inhibit phosphodiesterase. I don't know why that's there. Here we go. Cholera toxin permanently activates the G protein, causes an overproduction of cyclic AMP, causing loss in water and ions from all kinds of stuff. So it can actually kill you because it opens up those channels and keeps them open. So here is the structure of inositol triphosphate, also known as IP3. Here is the structure of diacylglycerol. So notice that this would be glycerol right here. You'd hydrolyze right here and hydrolyze right here. Okay, this other alcohol is where the IP3 was bound, that was cleaved by the enzyme. So here is this that would be stored in the membrane, right? So that gets phosphorylated by ATP, the enzyme that does that. And then when phospholipase C is activated, it will cleave this right here. And when you do that, you get IP3 and you get DAG. Okay. So I tried to put into words what does IP3 do and what does DAG do. So here's another scenario. So before I could ever find this, I had to draw my own. Maybe I don't even need that other one now. But anyway, so here's your first messenger, hits the receptor protein. Here's G protein that gets activated, right? Then it goes to phospholipase C and phosphorylates it. Now it's active. So it takes the lipid that contains the DAG and the inositol triphosphate and cleaves it. That's what lipases do. They hydrolyze. So the DAG goes over here and activates protein kinase C. And what does protein kinase C do? It's a phosphotransferase. It's a kinase. It simply puts phosphate on proteins, whatever proteins happens to be in that particular cell. That's what it does. It simply phosphorylates proteins. Different cells will have different proteins in those cells, and so the response to that cell will be different. What does inositol triphosphate do? It binds to its receptor protein over here in the endoplasmic reticulum and opens up this channel right here, which causes calcium to come out into the cytoplasm. Okay, then what happens? It binds to a protein called calmodulin, and calmodulin can activate another protein kinase. And what does that do? It puts phosphate on other proteins that may either turn on an enzyme or turn off an enzyme, depending upon what proteins are in that particular cell. The calcium may also, as free calcium, be used to do other things as well. So we're not concentrating on what the response is of that particular cell. What we're concentrating on is this mechanism of how the first messenger creates the second messenger and causes the metabolism of that cell to change. That's what we're focusing on. Okay. So any questions so far?
No. Too much.
It's too much? Yeah. Think about the people who had to figure this out.
That's true.
So when I got hired at the Cancer Center in 1986 or 7, something like that, I worked on the inositol phosphate problem. And I didn't realize when I first started that, I was competing with huge laboratories with Nobel Prize winners and all this kind of stuff. And anyway, I'm just by myself in the lab working for my boss over there. And yeah, but at least I was in the game a little bit. I was kind of barking up the wrong tree in the wrong forest on the wrong planet, but that's okay. So that's the reason why I have these old drawings. Okay, so here's another second messenger system. And so, where are we? So here's a typical scenario. So you get 1,8 cyclase producing cyclic GMP, which is a messenger in here. And what it does is causes the release of glutamate and the release of calcium. So glutamate comes over here, binds to the receptor proteins over here, and allows calcium to come in here. So here's the first neuron, here's the second neuron. So the calcium comes into the cell. It now binds with calmodulin. Calmodulin is a protein that modulates the concentration of calcium in a cell. It can also be used as a signaling mechanism. So the calcium in the calmodulin protein goes to this other enzyme called nitric oxide synthase. Well, what does nitric oxide synthase do? It takes arginine and converts it into citrulline, and it ends up releasing nitric oxide. Okay, what does nitric oxide do? Actually, two things. It does the same thing, but in different cells. Because nitric oxide is a small molecule, it's rather polar, and it can simply diffuse through membranes. So now you produce nitric oxide, it's diffusing in every direction. But within this particular nerve cell, it goes to guanylate cyclase that takes GTP and converts it into another messenger, which is cyclic GMP. And what does that do? It causes the release of glutamate. Okay, but glutamate was the major receptor over here. But when nitric oxide diffuses upstream, what does it do? Oh, it releases more neurotransmitter. So we call this a retrograde effect. This cell, when it starts producing nitric oxide, not only causes the release of neurotransmitter from itself, but it goes upstream, causes the release of more neurotransmitter from presynaptic nerve cell. What it does essentially is it supercharges this nerve transmission right here. Okay, well, let's go to this again. So here's the scenario, right? So first neurotransmitter is glutamate. Glutamate releases the synapse. It binds to the receptor. That's a calcium channel. Calcium enters the nerve cell where it combines with calmodulin. The calcium calmodulin complex activates nitric oxide synthetase, which essentially does this. And notice this reaction right here. Very unusual. This is an oxidizing agent. This is a reducing agent. So you're both oxidizing and reducing at the same time. That nitrogen in the amino acid arginine gets oxidized to nitric oxide. At the same time, the rest of the amino acid is being reduced by NADPH. So arginine is found in the amino acid pool. It simply means everywhere in your body, including proteins. Citrulline is another amino acid, but it's not found in proteins. It's not incorporated into proteins. So nitric oxide is involved in the relaxation of smooth muscle, vasodilation, which then lowers blood pressure, lowers inflammation. It also inhibits glycolysis, platelet aggregation, and recently we found it was a neurotransmitter in the brain. So it's carried by red blood cells and albumins in the blood, and that helps individual cells push their way through capillaries. Capillaries are so small, they're actually smaller oftentimes than the red blood cell itself. So the only way the red blood cell can get through there is if it relaxes the tissues around that. And so that allows them to get where they couldn't get before. Okay. So then nitroglycerin was used to treat angina. Angina is when the smooth muscles of your chest get extremely tight and you have a hard time breathing. People think that they're having a heart attack. So nitroglycerin is the same nitroglycerin that would be used as an explosive, but that's in its dry form. In its wet form, it's used as a way to relax smooth muscle. So then they were looking for alternatives to nitroglycerin, and they discovered, by accident, Viagra. So Viagra and its counterpart Cialis they inhibit phosphodiesterase, enhancing the effects of nitric oxide. So those two drugs act just like nitroglycerin, and they activate nitric oxide. And so whatever, people should not be taking nitroglycerin and Viagra or whatever at the same time, because they do the same thing. They could cause an overproduction of nitric oxide. And also you need to be careful of people who like start taking arginine, because they think that's going to do the same thing as Viagra. So combining all these things can be dangerous. So there's other things in food that can produce nitric oxide, might be responsible for the headaches that you get from MSG. Nitric oxide is also released by brain tissue during a stroke. Anyway, and now they're studying carbon monoxide. They believe that carbon monoxide may also be a retrograde messenger. So carbon monoxide is a toxin. It's from air pollution, from incomplete combustion of fuels, essentially. Same with nitric oxide. So these are components of smog, of air pollution. And so when they first started measuring it in nerve tissue, they're like, oh, we gotta make sure we're not, the lab isn't being exposed to a toxic environment from outside. And then when they fixed that, they found that, oh my God, it was still there. And it had biological effects. Anyway. So any questions about any of that?
No.
So this part in a red box, I just thought that I would just go into something as a medical example and to show you how complex it is. And, you know, I would never expect you to be able to know this stuff. If you did, you'd be a cardiologist and you'd be driving a fancy car and you know, all the other things that cardiologists have money to do. So about 1% of your muscle cells in the muscle, heart muscle, can generate electrical impulses or action potentials. So these are called pacemaker cells, and they directly control the heart rate. So when the pacemaker cells become unsynchronized, this is where a defibrillator is useful. What does a defibrillator do? It simply depolarizes all of the pacemaker cells, and then they all come back together in unison. So if you watch TV, all those hospital things and everything, they're all using defibrillators for almost everything. Oh my God, so-and-so's depressed or something, then they get out the defibrillator. They solve every problem with the defibrillator. But it's really only when the pacemaker cells have become unsynchronized. That's what you're trying to do, is simply resynchronize them. So you have primary, secondary, and tertiary pacemaker cells. So the primary is a group of cells. They would make your heart go about 100 times a minute. So the rate is actually modified by the sympathetic and parasympathetic nerve fibers, so that on average you get down to about 100 beats per minute. 70 beats per minute. Mine is more like 60. I know some real athletes, they're in the 50s heart beats per minute. If you have a large muscle and you're not like a huge person, you don't need to go 70 beats per minute. So what happens if the primary pacemaker has an issue? Well, then the secondary pacemaker can take over. So that gives you about 40 to 60 beats per minute. If that isn't working, your tertiary pacemaker, which is in a different part of the cell, gets you down to about 30 to 40 beats per minute. And if you would look at the evolution of the heart, this is indicating the evolution of the pacemaker cells. So some people can get their heartbeats down to very, very low amounts by concentration, and they've learned to manipulate their nervous system to do that. And maybe you've heard of people that they were drowned, they were dead they were underwater for like 20 minutes and then they revived them. So all this has shut down, but the tertiary heart pacemaker was still able to keep that person alive. So when they reactivate the heart by whatever means, maybe defibrillator would be good in that case, then you can oftentimes bring this part of the heart back and the people revive. So how do you look at people's hearts? Well, you do the treadmill stress test, electrocardiogram, all that kind of stuff. In that case, you'd be looking at the different phases that go through. But when it comes to treating heart disease, there's really only 6 classes of heart drugs. So here's some of the things that treat hypertension. So these are angiotensin converting enzyme inhibitors. Okay, angiotensin 2 receptor blockers. So you inhibit the production of angiotensin, or you block the binding of angiotensin to its receptors. There's also renin blockers, there's alpha blockers, alpha agonists. So they interact to decrease adrenergic neurotransmitters from getting into their receptor proteins. And then you have antiarrhythmics when the heart rate is too slow, too fast, or irregular. So we have beta blockers, and you have calcium, potassium, and calcium ion channel blockers. These are all the different kind of drugs that you have. To lower cholesterol, you could use statins. Blood thinners would be like aspirin, heparin, warfarin. Heart rate control would be digitalis, which we'll look at. And vasodilators are like nitric oxide, either from nitroglycerin, Viagra, or Cialis. So those are the classes. And one thing that's rather interesting is that these ACE inhibitors, this turns out to be the receptor for the coronavirus. Okay, that's how it gets into the cells, via the ACE2 receptor, not the ACE1 right here. Anyway, so what the heck is angiotensin and all that kind of stuff. Well, here's a good map of what it is, okay? So here's the liver. It produces angiotensinogen, and then it gets converted into angiotensin 1 by an enzyme. And here's where the enzyme, another enzyme is, where it gets converted into angiotensin 2, which then goes and acts as a hormone, is the primary effector in all these different issues over here. So if you inhibit this enzyme, you stop angiotensin 1 from being converted into angiotensin 2. If you inhibit this enzyme, then you inhibit the whole kidney part of it, okay? So beta blockers, ion channel blockers, here's an example of that. So here's the G protein gated ion channel that we've seen several times before, okay? And so these are ions, and so what we're trying to do is block in these several different ways over here. Okay? So you're messing with this signaling system over here. So ion channel blockers block G-gated protein ion channels. So what do you mean by G protein gated? This thing can't open if you inhibit any part of this process over here, okay? So this opens and closes the gate being the ion channel, okay? So statins we've talked about before. Aspirin inhibits the enzymes that produce thromboxane, the prostaglandin that promotes platelet aggregation. Here's heparin is a blood thinner. Warfarin also inhibits clotting because it inhibits the synthesis of platelets. Okay. And digitalis really refers to cardiac glycosides. So digitoxin is extracted from the foxglove plant. And anyway, so here's some drugs. So do they have actually digitoxin? No, this is the genin over here. But anyway, so digoxin is widely used as treatment for various heart conditions like atrial fibrillation, atrial flutter, and they have different drugs that are slightly modified. Okay. So what if one of your friends told you, "Oh, I've been drinking foxglove tea. They say it does this for my body." Maybe it's foxglove, maybe it's some other herbal tea. Do you think that all those herbal teas are natural and then would have no negative effects on your body? Or do you think many of them might have some really profound biological effects that you could be injured by? Anyway. So anyway, growth factors. Growth factor is a protein or hormone that stimulates cellular growth, proliferation, and any of these other type of things, right? So human growth hormone is being used in all kinds of athletes and everything else. And yeah, it's really kind of interesting. So one of my son's friends is a pretty big guy. I think he would be big anyway, but he has a what's the word for it, a vitamin shop. And anyway, I'm a little bit worried about them taking all this growth hormone stuff. And pheromones is a molecule that's secreted that triggers social responses in members of the same species. Okay? And so there are people who sell pheromones. I see them advertising in the back of magazines like Discover and Scientific American. But as far as I can tell, there's no evidence for pheromones working in humans. It does not stop people from buying them and making money off of them. But pheromones are known in the insect world especially. So here's some pictures of some of them. So let's see. Yeah, anyway. So they're claiming that these are acting as pheromones in humans, but they don't really have any strong evidence for it, as far as I'm concerned. Now, bisphenol A, I just thought that you should be aware of this. So bisphenol A is found in several plastics. Okay? That's why I would never microwave any food in a plastic, because I don't want bisphenol A to leach into it. So here's bisphenol A, how they make it into a polymer. Okay? And so does this mimic these hormones? People claim that it does, other people claim that it doesn't. So here's a statement that comes from the World Health Organization, just to let you know. They're finding BPA in small amounts everywhere in the environment because plastic is everywhere in the environment. But the World Health Organization still maintains that there's such low concentration that they're not causing any problems. So RNA has been known now to be a messenger. There's different types of RNA. So some has been found to be involved in long-term memory in snails, mice, and rats. Yeah, so it could very well be another type of messenger. Exercise, I keep talking about exercise. Here's some of the latest things on exercise. So what we have found recently is that the hippocampus keeps generating neurons throughout your life. I was just explaining this to a group of friends the other day. Oh, I forgot to do that, I'm getting old. It's like, yeah, you're getting old, but you know, your brain getting old is not an excuse, because we know that we do keep generating neurons, at least in the hippocampus. And what they've found in other experiments, in older adult humans over a 1-year period, aerobic exercise led to an increase in brain-derived neurotrophic growth factor. That's a growth factor right there for the brain. So the size of the hippocampus increased and the number of neural connections increased. So there's also a correlation between sleep and neuroplasticity. So this theory, synaptic homeostasis theory, suggests that during the night when we're sleeping, we prune, and then we prune some neural connections, and then we strengthen other neural connections. So it's like whatever you have experienced during the day, you cannot keep track of everything. You would be overwhelmed. So in unimportant events, those kind of neural connections are pruned when you're sleeping. Important events that might have to do with the survivability of you are then strengthened. So what we do at nighttime is we reduce all the things that would basically make us confused. You ever notice if you don't get a good night's sleep, the next day you're kind of dragging around, you're easily confused, that's your problem. You didn't go through that pruning process of the connections that you made during the day. So that's what neuroplasticity is. You have to be able to break and form these connections so that you can learn from your life, so that you can increase your survivability. And anyway, stress affects that. And here's a little bit about short-term stress and long-term stress. And cortisol is the stress hormone. It perpetuates stress by activating the hypothalamus to release more of these hormones that are involved in stress effects. So your body has a stress response because that is supposed to help you run away from a predator or do something stressful, but it helps you get that particular task done. But you don't want your body to be in a constant state of stress. Anyway. And I put some stuff in here about chocolate because I just thought it was interesting. I think everybody loves chocolate. And I found this information about Hershey's chocolate. I thought that was really kind of interesting. So I thought I would talk to you about it. And then brain size and intelligence, they don't correspond. So there's birds that can do tasks that require intelligence at the level of chimpanzees and primitive humans. How do they do it with these small brains? And then they don't even have a cerebral cortex of any extent. So what this group did is they didn't look at just brain mass, but here's brain mass over here, but they looked at the concentration of neurons, basically the neuron numbers within a certain, in their brain. And when they do that, look, here's humans. We're still ahead of elephants, but look who's right behind them. Crows. Okay. So not all birds are smart. I mean, like here's a chicken over here. They're not very smart. But everybody talks about a pig being so brilliant, right? And I have a friend who's an expert on pigs, and she basically coerced my French colleague and I to look at pig brains. And she said pigs are the smartest of the animals because they can pass the mirror test. And the mirror test is if you put something on their back where they can't reach it, they will actually go up to a mirror and see what's on their back. And so my first response is, yeah, my wife can pass that test. But the pigs are not even as smart as many of the other bird species. So with humans and elephants and pigs, we look at their brains and we seem to understand how their brains are structured, but that doesn't correlate necessarily to intelligence. So anyway, yeah, serotonin and dopamine, really the only two things you ever enjoy. So here's norepinephrine, serotonin, and dopamine. And they do all these things, but what's all in the middle right there is mood. So just some other cartoons over there that, yeah, I still love this one. That one is just, yeah, that's pretty funny. So that's it. Anybody have any questions on anything?
No, not at this time.
I know it's overwhelming. And, you know, in one sense, I'm giving you all this information because I want you to see the utility of it. I want you to see why this stuff is important to know. But what I really need you to know, what I'm going to test on, is the basic stuff, okay? Not the applications. It's the basic signaling, the basic concept of a neurotransmitter and a hormone and all that kind of stuff. But when we review for Unit 4, then we'll get into all that stuff. Okay? What's important and what's just enrichment. So you guys ready to have a nice weekend then? Okay, well, you guys have a great weekend, and I will see you Monday.
Okay, thank you.
You take care. Bye.
Bye.